So far through our discussion of immune system, we focused on the innate, the non-specific side, and we've looked at the humoral side, the production of antibodies by plasma cells. So now we're going to look at the last type, and this is the cell-mediated immunity, and this is where the T lymphocytes come in. Now we call them Ts because they become immunocompetent in the thymus. And when we say this immunocompetency, I want you to realize it's extremely important because with cell-mediated immunity, much like the rest of the immune system, it's critical that these cells do not recognize your normal tissues and normal receptors. So it's in the thymus that we are going to largely prevent ineffective T cells from escaping out into the systemic circulation, and therefore doing our best to prevent an autoimmune response. We're going to see that most of the T cells that begin to form never even make it to the final developmental stage to be part of the immune system. Now the other system, or the other thing that makes T cells distinct from the other cells that we've seen, is that T cells aren't directly contacting antigen. Macrophages, they can go gobble stuff up. We can see that many of our granulocytes can recognize pathogens. B cells have receptors for antigens on their surface. However, T cells must be presented an antigen by an antigen-presenting cell like a macrophage or a dendritic type of cell. And so we're going to see that these antigen-presenting cells are going to display antigen on molecules that are called MHC markers, or major histocompatibility complex markers. And so our T cells are going to have receptors for the combination of antigen and specific MHC molecule with which the T cell can then become activated. In this illustration, we start in the upper left. Number one, we see an antigen-presenting cell in the skin. We refer to them as dendritic cells. They're phagocytosing a pathogen. They're displaying an antigen on their surface. And what we then see is that these cells migrate then into one of our lymphoid organs, where our lymphocytes are going to reside. This is a lymph node. We have a high population of T lymphocytes as well as Bs. And it's within the lymph node that this dendritic cell, MHC antigen complex, binds with an appropriate receptor that can recognize that antigen. And then this particular T cell becomes activated. Again, it's within the lymph node. However, that activation is going to start a chemotactic reaction in which that T cell then diapedeses out of the circulation and into our tissues, where it can direct cellular toxicity, and it can destroy the pathogen out in the tissues. So a little more about these major histocompatibility complex molecules. We have two of them. Type 1 and Type 2, or Class 1 and Class 2. I like to think of 1 as most important. We're number 1. You hear that in all sort of athletic events and competitions. So the Class 1, you're going to find this on every cell surface of every cell in your body except for red blood cells. And this Class 1 MHC can only activate a subpopulation of T cells called cytotoxic T cells. We're going to see this is extremely important for cells that become cancerous, tumorigenic, or virally transformed. And so some of those markers are going to be displayed on these Class 1 MHC molecules for the corresponding receptor on T cells to recognize and know, okay, this is a tumor cell, it's infected with virus, we need to kill it. That's why it's on every cell in the body. except red blood cells. Now, this is distinct from a class 2 type of MHC molecule, which we find primarily on those cells we refer to as antigen-presenting cells, or we abbreviate them APCs. These class 2 MHC molecules are used to display antigen and present it to the receptors on helper T cells. Now, the receptors on our helper Ts can only recognize class 2, and our receptors on cytotoxic Ts can only recognize class 1. It keeps them distinct. And so we introduce those receptors in this illustration. Our helper Ts are able to recognize class 2 MHCs because of their CD4 receptor. So T cells are said to be CD4 positive T cells. Whereas our cytotoxics, or sort of loosely termed killer T cells, these recognize the class 1 MHC with their CD8 receptor. So these are the combinations that we have for our cell-mediated immunity and its activation. Now, I like mnemonics. I like tricks. Unfortunately, I couldn't come up with a mnemonic for these pairings. It seems fairly straightforward, but I challenged a class a couple of years ago to see who could come up with a mnemonic, and I would give them extra points. So this is not my fault, what I'm about to say. It's the best we could do. All right? Juan ate with 2 forks. I know it's bad, right? But it's so stupid and so corny, you can't forget it. It's like you can't look away from a train wreck. Juan, the number 1, ate with 2 forks. And that's how you can keep them linked together. So 1 goes with 8 and 2 goes with 4. So let's look at how these T cells become immunocompetent and yet how we screen them to make sure they cannot recognize self and cause an autoimmune response. So the T cells derive from stem cells in your bone marrow. They're transported to the thymus where they are going to complete their developmental stage. And the first thing they do is they go through a positive selection step. Now, in the thymus, we have the cortex and medulla. Cortex on the outside, medulla toward the middle. And these cells are going to start to develop from the outside in. And when you get to the medulla of the thymus, that's where the blood vessels are going to be that you can use to transport away from the thymus. We also have cells in the thymus that are called reticular cells, and they're going to form barriers. So they're going to functionally and, in fact, structurally separate these compartments. So as we start in this outermost compartment, we're going to have these developing T cells, and we're going to have antigen-presenting cells. And these antigen-presenting cells are going to present antigen to our growing immature T cells, and only those T cells that can adequately bind so that we know those T cell receptors work, only those cells are going to be allowed to progress to the next compartment. If the receptor doesn't work, if it doesn't bind strong enough, or if it in fact binds too strong, those cells are going to be instructed to undergo apoptosis, or basically kill themselves. Positive selection. And so now the cells that have receptors that work move closer to the medulla into the next compartment. And here we undergo negative selection. And in the negative selection step, what we're in fact doing is the antigen-presenting cells are challenging the T cells with self-recognition markers. So only those T cells that have functional receptors have gotten in. Now only those T cells that do not recognize the self-antigens are going to be able to pass through and make their way out to the circulation. If, in fact, a T cell recognizes self, that antigen-presenting cell is going to instruct that T cell to undergo apoptosis. So your receptor has to work, and it has to recognize something other than yourself. And in that case, what you end up with are about 80% of the T cells that begin developing undergoing apoptosis. You have a ton, because remember, just like we use the variable region and we somatic hypermutation so we could recognize any particular antigen, T cells have a lot of variability going into their receptors as well. So we have a huge number, but we have to make sure they work. And because of that random variability, they're going to recognize your own cells and tissues. You just don't want them to get out in your circulation once they begin to be immunocompetent. So that's why you kill so many, so you can have the variability and still be safe. So this illustrates our positive selection in the cortex. As we move deeper into the medulla, this represents our negative selection, and you can see the association of our antigen-presenting cell in both. First step, that association is a good thing. The second step, it's a bad thing. So that's how we ensure T cells are properly functional when they get into our circulation. So you've already learned we have two types of T cells. And the first that we're going to look at in more detail are the CD4 positive T cells. You're going to hear me say those CD numbers a lot to get you comfortable associating those receptors with a particular type. So the CD4 positives are our helper T cells. Now, our helper T cells are going to be like the dispatchers at the police station. Because when these antigen-presenting cells come around and stimulate that helper T cell, the helper T cell is going to be secreting chemical signals to activate many other members and components of the immune system. Not just other members of the specific immune system, but also cells of the innate immune system. So the helper Ts call the immune system as a whole to activate. And they do that through our chemical signals called cytokines. Sometimes they're referred to as chemokines or lymphokines. They're our chemical signalers of the immune system. Now, even though we have two different subtypes, don't worry about the subtypes of T cells. And you see it in this illustration where you see our Th2 and our Th1. So these different subtypes are secreting these chemicals, IL-2, IL-4. These are our interleukins. We're going to talk about other cytokines and their specific functions. But you can see in this case, a helper T is activated by our antigen-presenting cell, and that helper T can go on then to activate other T cells and produce more memory T cells or more activated helper Ts that secrete the cytokines. And in this case, our interleukin-2 and interferon, they're interacting on a monocyte. Monocyte becomes activated, begins to clone itself, migrates out into the tissues to take care of the pathogen. And once the monocyte leaves the circulation, it changes its name, right? What's the new M name for a monocyte in our tissues? Macrophage, a macrophage. But you see, other T cells, they can go on to activate B cells, part of our humoral immune system. And once these chemical signalers start the cloning of our B cells, now we're also going to begin to produce antibodies that will then recognize that particular antigen. Now, our CD8 positive B cells, cytotoxic Ts, we've already been talking about how they're going to interact with cells that are virally infected or becoming tumors. And CD8, remember, is going to be recognizing antigen presented to it by MHC class 1s. Now, the way our CD8 cells kill these infected cells or cancerous cells is by producing chemicals that are kind of like granzymes. These are enzymes that can be secreted across space and can begin to break down the cell. I like to think of it like a hand grenade. They can also create and secrete proteins that form pores, much like our attack complex and our complement cascade. These protein channels that insert themselves into the plasma membranes of the damaged cell, and it loses its cytoplasm and dies. But we can also send chemical signals from the cytotoxic T that causes caspases to become activated in the damaged cell. And remember, caspases are enzymes that are part of the process of apoptosis. So there are a number of ways that cytotoxic Ts directly, once they contact that MHC1 and antigen, directly lead to the death of the infected or the damaged cell. Scientists love to categorize things. I think you've probably learned this coming through all your biology courses. We like to label it, we like to put it in the right place, organize it. And so when we find cells that don't exactly fit, we create a new category. So here we have cells that are called natural killer T cells, which is easy to confuse with cytotoxic Ts because they both kill. Well, in this case, our natural killer Ts can become either helper Ts or cytotoxic Ts. But the reason we put them in a separate category is because they have an additional receptor molecule that is presented to them on a CD1D, not a class 1 MHC, not a class 2. So since there is a difference, we think of these as sort of an intermediate between the helper T and our cytotoxic T, but they can become either. So it's almost like a reserve of our cells that haven't quite finished their developmental pathway yet. Now, our fourth type is one that you may think, why would we have this? But when we think about homeostasis in the body, remember one of our predominant and dominating controlling mechanisms is negative feedback. And when you look at antigens being presented to helper T secreting chemical signals that cause our immune system to ramp up, other immune cells secrete these cytokines and they're going to ramp up. That looks like a positive feedback. We have to have a way to shut it down and to keep it from getting out of control. And so our regulatory Ts that were formerly called suppressor Ts, and again, I add that name there because that's what they do. Regulatory Ts do not enhance a T cell mediated response, but they in fact suppress it and help turn it off. So these cells are going to begin to divide and grow over time when you activate a T cell response, and as they do, they are going to begin to kill your other T cells. Just like our clotting cascade, fibrin gets stickier and stickier and you create more and more and more. But remember factor 12 that started the intrinsic pathway? It also activated kallikrein, plasminogen activator, led to the activation of plasmin, which destroys a clot, but it's a time delay. Regulatory Ts are on a time delay, so as you get more T cells taking care of the infection, you finally have activated enough of these that they're going to stop the T cell response because you have to have a way to shut it off. Now, you may think that, okay, well, T cells are doing a good thing, and if you don't shut them off, or if they get too busy, is that really a bad thing? And the answer to that is yes. And it's exemplified in the simple fact that when you look at regulatory Ts and their receptors, in addition to CD4, which is similar to helpers, you also see this receptor FOXP3. And if you happen to mutate FOXP3, just that one receptor, this can lead to a disease that is referred to as IPEX. Now, if you think you had a cold and you felt really bad with a cold, you definitely do not want IPEX. Listen to the name of IPEX. Immunodysregulation. If you lose FOXP3, so you lose functioning of your regulatory T's, your immune system is jacked up. Is that bad enough? No. Polyendocrinopathy. Your endocrine system is jacked up. The endocrine system regulates everything in your body. Add on enteropathy. Entero is your digestive tract. Montezuma's revenge to the 100th degree. So, yeah, just that one receptor on the regulatory T that suppresses a T cell response can totally ruin your day. So they're extremely important. So here's our regulatory T. We have a T cell that's undergoing its response. When they get together with their receptors, boom, it's going to lead to apoptosis of our cytotoxic and or our helper T cell, whichever one the regulatory T is binding to. And that's how we stop a T cell response. Just as B cells, when they clone, become plasma cells that produce antibodies or memory cells that can be recalled at a later date with a subsequent exposure to a pathogen, T cells also produce memory cells. And CD4 positive T cells produce CD4 positive memory cells and so on. The same memory cell can't become either. It has to be on a certain lineage. So you've got a memory for your cell-mediated immunity, just as you have a memory with your humoral immunity and your B cell lineage. So in our illustration a few slides back, we saw our helper T cells secreting IL-2, interleukin-2, which is one of our cytokines. And we have several cytokines. I think we're going to list maybe about 6. And what I want you to do, especially what you see underlined or in bold, is what I really want you to remember with these cytokines. Interleukin-1, this is going to be a principal cytokine that induces fever. Now remember, all those positive things we said happens to your immune cells when you increase the temperature. Well, that's what we're doing. We're increasing our temperature. Remember, fever was part of the innate immune system, but in this case, it's enhancing T cells as well. There's interleukin-2 that we saw in our slide. Interleukin-2 produced by helper T cells. They activate cytotoxic T cells. And what's interesting is there's a drug, proleukin, which is an synthetic interleukin-2. It's often used to enhance chemotherapeutic agents when you're treating cancer patients. Here we have interleukin-4. Interleukin-4 is going to be produced again by helper Ts. It activates B cells to produce antibodies. And in fact, IL-4 is extremely effective at causing our isotype switching of whatever the antibody that B cell is going to produce, switching to an IgE type. So interleukin-4 is fairly busy when we're starting an allergic response and reaction. Interleukin-5, we're not going to go through all of them, we're just touching on these 6. Interleukin-5 produced by helper Ts, and you may see some of this in mast cells. So once again, it's not just the specific immunity with our T cells. Interleukin-5 is going to stimulate B cells to grow. It's going to cause eosinophil activation, and since we're talking about eosinophils, granulocytes, it's probably also going to interact with B cells as well, because you see it on the increase during allergic reactions and allergic responses. You'll see interleukin-5 produced. So our last two, interleukin-10, It's going to be an anti-inflammatory cytokine, which is almost anti-intuitive because remember, in an infection, mast cells are going to produce a ton of histamines, heparins, which lead to inflammation. Remember, when we have antibodies opsonize on a pathogen, that's going to start the complement cascade. What's one of the effects of C3b, C4b? Increase inflammation. So here we're going to shut down inflammation. But you see how we've got that negative feedback so that our immune system doesn't run on at high speed forever. So it's going to be important to slow down an anti-inflammatory response. And again, what cells make it? Monocytes, macrophages, lymphocytes, mast cells, which is interesting because the mast cells are the one that cause the inflammation in the first place. They're going to have a mechanism to shut it down as well. So that's part of the negative feedback. Interleukin 12, this is interesting because it is produced by dendritic cells and macrophages. Remember, those are two of several of our antigen-presenting cells. So not only do these antigen-presenting cells display the antigen, they also secrete IL-12, which causes our naive T cells that haven't quite become immunocompetent yet. It causes them to grow up to the point that they can recognize antigen. So it's going to recruit more T cells to a developmental stage where they can actually recognize the antigen that these cells are actually trying to present. Maybe this is happening inside of a lymph node or another lymphoid organ itself. That's a lot we went through, right? 6 cytokines. But when you look, as you see in this illustration, that's not even the tip of the iceberg of all the chemical signalers that are going on between and back and forth in this two-way communication. We got through IL-12. That was the last one. And we skipped a lot, right? Look, here's IL-16. There's even more. We've got transforming growth factors. We've got tumor necrosis factor. There's just tons of these chemical signalers. You can make an entire research career in studying one of these and its effects. The human body is an amazing machine, and the more we know, the more we realize we don't know and we don't understand because it's almost infinitely complex. So shutting down a T cell response is so important that we have regulatory Ts that do that. Well, there's another way in which we shut down a T cell response, and that's by causing T cells to produce a substance and secrete it into their local environment called the Fas ligand. Now, a ligand is something that binds to a receptor. And so this is called the Fas ligand because the Fas receptor was identified before whatever bound to it was identified. And the Fas receptor is on the surface of our T cells. So in an autocrine sense, as T cells divide and as T cells grow, they're producing Fas ligand. And whenever you get enough T cells produced that have secreted the Fas ligand, that you have surpassed a threshold concentration, the Fas ligand binds back to the Fas receptor. And when it becomes activated, look what you've got in the cytoplasm, that FADD. You remember what that DD stood for? The death domain. And it starts apoptosis. So basically, you can think of the T cells as they're doing their job. They're secreting T cell poison into their environment. And when there's enough T cell poison around them, which is about the time they've done their job, then they die. And you stop the T cell response. That's why you don't continue to feel bad after you've gotten rid of the infection or the pathogen. So those are the players, the innate immune system, the Bs and T cells of the specific immune system. Now let's see what happens when you actually get an infection. The innate immune system didn't work. The pathogen got through the skin. It got past our monocytes, our granulocytes. And so what happens is when we have B cells and T cells that recognize pathogen, the very first time, that is called a primary immune response. And in the case of B cells, they're going to begin to produce IgM, but over the course of 2 weeks, they're going to class switch to IgG. And so you're not going to feel good for about 2 weeks. But then you have enough antibody in your system. Your specific immunity has basically taken care of the infection and you begin to feel better. Like you do with a kind of cold that we get periodically. Now let's say that was chicken pox. You don't get to experience the fun of chicken pox anymore. But it's 2 weeks of just torture, itching and fever and just not feeling good and these ugly welts all over your body. Our loving parents back in the day, since we didn't have the chickenpox vaccine, and it's much easier for children to endure chickenpox than adults. It can be fatal for adults. Ask somebody that's had the shingles how much fun that is. So guess what our parents would do when they would discover there was a child in the neighborhood that had chickenpox? They would have a chickenpox party. And let's go play at little Susie's house. And we would all go play over there and we'd all get the chickenpox at the same time. You know what some people are doing these days? They are selling candy that their child that had chickenpox has licked or had, and people are purchasing this instead of getting a chickenpox vaccine for their child. Blows my mind. But the point of doing that is so you can go through these 2 weeks, and in those 2 weeks, you're producing IgG antibodies, but you're also producing memory B cells. So that when you're older and you do come into contact with the varicella zoster virus, again, that leads to chickenpox, now you mount a secondary immune response. Vastly more memory cells than you had B cells to begin with. And once you activate that memory cell, you're going to begin to produce antibodies, not in 2 weeks, but in 2 hours. And you are going to produce a ton of antibody. As you see in this illustration, look at the little bump of antibodies in a primary immune response after 2 weeks, and look at the huge jump in the antibodies that are produced in 2 hours. You've been exposed to chickenpox probably so many times, but you never knew it because your secondary immune response wiped it out. And so this whole concept is where we get our immunizations. Our whole idea of immunizations. Now, passive immunity. Remember, we already talked about passive immunity with the IgGs that cross the placenta, mom giving the antibodies to the baby, getting it in the colostrum, the breast milk. You don't have the cells to make it yourself. You're getting these ready-made antibodies from mom. Now, if you do this clinically, let's say if you have been bitten by a venomous snake, you're going to get anti-serum or antitoxin. You're getting antibodies that were made in some other mammalian species. They're going to inject you with those antibodies so the antibodies can bind the toxin and the toxin's not free to do damage to your body. That is passive immunization, getting ready-made antibodies. So mom can provide it to the baby, or the doctor can give you a shot to provide it. But what we're most familiar with are our clinical immunizations or vaccines. We just talked about the chickenpox. You all had a chickenpox vaccine. You probably all had the meningitis vaccine. That's to bolster your memory cells so you don't get the disease later. Now, depending on how effective that vaccine was, you may have to go back to the doctor every couple of years and get a what? A booster. What are you boosting? Your memory T cells. Now, supposedly, if there is a terroristic attack and they use the smallpox pathogen, supposedly I'm going to be okay. Because I got that vaccine when I was a kid. Y'all are in trouble. Because they stopped doing that a long time ago because smallpox has largely been eradicated, but smallpox still exists. They lined us up like cattle. I will never forget this. It was first grade. I didn't like shots anyway, so I was nervous. Lined us up, opened the door. One person went in. They shut the door. And you hear this sound. It was almost like a pop. And then the door opens and the kid comes out holding their arm. You go in and they have a gun. It looks like this futuristic silver metal gun, and it's got all these little needles on the end of it. And it basically pricks you fairly deep. And they put it on the back of your shoulder. Have you ever seen someone with that round scar right there? That's the scar from the smallpox vaccine. And what you're doing, once again, you're artificially stimulating a primary immune response to build the memory cells so when you encounter that again, you don't get the disease. Now, we don't always call them immunizations. We often call them what? Vaccinations. Do you know where we got this word vaccination from? Vacca means cow in Latin. And back in the day of smallpox, this brilliant doctor said, you know what? The women that are milking the cows don't die. They get the pox, but they don't die. So he surmised that the disease in cows, which is called cowpox, similar but different virus, somehow protected the milkmaids. So he got a needle. This is kind of gross, but he got a needle and dipped it into the pox of the cow and scratched it into the skin of some people. They got the pox, but they weren't as bad. They weren't as sick, but it prevented them from getting smallpox. So that became the first immunizations or vaccinations because it started with cowpox and smallpox.